Uh, hi, hi, everybody. I just want to thank all of you to be here. I just want to thank the time you're giving me to hear the the screens I'm going to giving you about the oncogenesis. Um, if you have any questions or if you have any problem with audio or you don't understand what I'm saying, just feel free to tell me or to ask me. Anyway, we're gonna have a set of some minutes at the end of this webinar to make any questions you have. Um, so then I'm going to share the this set of expositions for you. If you have any problem, just feel free to tell me. Well, look, we are going to talk about oncogenesis. What's oncogenesis is how cancer begins in the body around many hallmarks. We are going to see any one of them through this exposition. Um, how changes for one healthy cell to a malignant or a cancer cell. I would like to start with this phrase. I really like this phrase, which is saying all cancers are thought to share a common pathogenesis. Each is the outcome of a process of Darwinian evolution occurring among cells populations within the microenvironments provided by the tissues of a multicellular organism, analogous to Darwinian evolution occurring in the origins of a species. Cancer development is based in two constituent processes: the continuous acquisition of heritable genetic variation in individual cells by more or less random mutation, and natural selection acting on the resultant phenotypic diversity. This is what you are going to see. This is the first thing you have to learn about cancer because, as you can see in,
Doctor, sorry, I
uh,
think I don't know if I am the only one, but and you can expand your screen because
I forgot it. Yeah, thank you.
thank you. Thank you so
Okay,
much. Yeah.
okay, okay. Sorry. Ah, After that, well, you have to learn this at first when you're trying to understand cancer. Cancer, actually, some books of medicine says that the only way to be sure that you will not have cancer is not to be born. So the only fact to be born is a risk factor to have cancer. According to this text, is thanks to the evolution of the origins, the evolution of humans, of animals. Then our cells are making evolution themselves, though that's why we have a risk to make cancer. So in the next, we I got these sheets from GloboCan. This is from the World Health Organization that recollects all the information of cancer around the world. That tells that the total population of the world is more than 7 billions, almost 8 billion people living in this world. The number of cases in this on the last year, 2020, we have 19 million people who got cancer only that year. There are almost 10 million people died on that year because of cancer, and of over 50 million people have cancer through the last 5 years. So, according to this evidence, you can see that almost 50% of the people who got cancer die because of cancer. So, according to the statistics in the world, we can see the first cancer or the most common cancer in the world is lung, then prostate, then colorectal, stomach, and liver. More than the 50% of all cancers in the world are in groups of only these five types of cancer. Lung at first. If you can see, obviously the most common cancer in women is breast cancer, but you cannot see on this table. If you can see these tables, you can see the difference between men and women around the world. In men, you can see the first one is lung, prostate, and colorectal, stomach, liver. the exact amount, not the exact amount, but the same types of cancer in all both sexes are the same types of more common cancers in men. So after that, we can see the table of number of cases in 2020 females, all ages. The first one is breast, then colorectal, then lung, then cervix, uterine, and finally thyroid cancer. Both tables groups or make groups of only five types of cancer. That only five types of cancer are bigger than the rest types of cancer in the world, more than 50%, actually 52, 53% in both groups, men and women. If you see this table here, you can see the population of men, the population of women in the world, and you can see the entire population here. The number of new cancer cases is 10 million in men, 9.2 million in women. Then the age, um, the risk of developing cancer before the age of 75 is 22% in men, 18% in women. But this is exactly what I want you to see is in males, lung, prostate, colorectal, stomach, liver. In females, breast, colorectal, lung, cervix, uterine, and thyroid. But if you can, please try not to memorize with taking care of this data, because then we're going to see the cancer in Mexico. We have to understand cancer in the world and cancer in Mexico. If you are trying to see patients, you're trying to make a better life for people with cancer. Well, the cancer in Mexico, the number of new cases only in 2020 was breast cancer, the first place, then prostate cancer, colorectal, thyroid, and cervix uterine cancer. The total of population in Mexico is almost 129 million people. The number of new cases is almost 200,000. Number of deaths, 90,000. And number of prevalent cases in the last 5 years is half million people just in Mexico. You can see the tables. If you can see the difference between around the world and in Mexico is enormous. It's amazing, the difference. Prostate in the first place, colorectal, stomach, lung, and the fourth place, and non-Hodgkin's lymphoma. Then in women, the number of cases 2020 was breast as the first place, cervix, uterine, thyroid, colorectal, and then corpus uterine. If you can see the some of the problems of the government in Mexico, you can see cervix uterine. People are dying of cancer in cervix uterine despite we have a vaccine. You can get a shot against this type of cancer. So, but the vaccine does not exist. It exists, but it's really hard to get it in Mexico. That's why people are dying every day because of this cancer, um, and many, many, many other factors. But this is why I wanted you to see or to focus on this data, because the difference in Mexico against the entire world is completely different. Okay, so just I promise is the last picture of data. The population of male is 63 million, females is 65 million. The risk of developing cancer before 75 years old is almost the same between men and women. Is in the median of uh, 14.3. And the five prevalent cases, and then again, I want you to see this data, this kind of cancer. Um, the most common cancer in Mexico is breast, prostate, colorectal, thyroid, and cervix artery. Almost exactly the cancers of the most common cancers in women in Mexico. This this you can see how or how women are having most cancer than men in Mexico. The data is almost exactly in this place than in this place, just because of colorectal between they both. But despite colorectal is in the both columns, men and women, you can see breast at first. Almost the evidence can tell that uh, between 11, 12, or 5 or 15 percent of women are going to have cancer. According to statistics, 50% die of cancer. So in Mexico, maybe 200,000 women are going to die because of breast cancer. So that's why you have to try to stop this, making a change in your patients or making a change in yourself on your own. So, okay, let's go to see. Um, hereditary, before this table, I just want you to notice that hereditary cancer means approximately between 10 and 15% of the entire cancers are hereditary. So this is how you are going to prevent. Just give it a number, for example, if 100 people are having cancer, you can just prevent 10 people from having cancer because 10 or 15% are hereditary. So how you get it? First, trying to once you understand immunology system, once you understand uh, molecular biology, then you can play to be God. Then you can play to develop new treatments, new diagnoses for a cheaper cost, of course. Okay, then sorry. Yeah, you can see here how you can have cancer, but hereditary cancer. So you have here, this is a passenger mutation. You have no mutation in the gestation when you are inside of the uterus of your mama. Then you can have a mutation. Then in the infancy, you are having this mutation again. Then is when you are in your childhood, you get exposed to smoke, to bad weather, to the smoke of the cigarette of your family. For many risk factors you're getting exposed, so you can have different somatic mutations. You have some mutations since you were born, but you have new mutations, then you are exposed uh, to some risk factors. Then, when you are carrying a mutation, this is a driver mutation. When you get one of those because of the environmental and lifestyle exposures, that means the blue color, and then the yellow color again a driver mutation, but the in-strict mutation processes. So 10% of the cancer is hereditary, but 90% of the cancer appears along. You don't have to have risk hereditary factors to have cancer in the 90% of all the cases. So this is a benign tumor, intrinsic mutation processes. Then you have cancers, a mutator phenotype that we're going to see in a few minutes what means a phenotype, what means a mutation. But you got the DNA is changing in the cell, so it's modifying the genome, it's modifying then the phenotype. and then it's creating a cancer cell. Then you have cancer, you are receiving chemotherapy, and then you have in yourself mutations, cancer, and chemotherapy, useless chemotherapy, because you can have at least 10 or 100,000 or more passenger mutations. This is the lineage of mitotic cell divisions from the fertilized egg to a single cells within a cancer, showing the timing of the somatic mutations acquired by the cancer cell and the processes that contribute to them. So this is the process to you to have cancer since uh, you were created, since you were born, since to when you have cancer, really complicated cancer, chemotherapy resistance, and then we are gonna see the common treatments for cancer, use the most usable cancer treatment for cancer. Then in the year 2011, Douglas Hanahan wrote the Bible of Cancer. This article became to be the Bible for cancer research. This article explains or reviews itself how cancer or what cell cancer has to do to become or to appear as a cancer cell, and then to establish for not to be killed by the human body, to be persistent and growing, and then to create invasion and metastasis. So the first step is creating sustaining proliferative signaling, then evading growth suppressors, activating invasion and metastasis, enabling replicative immortality, inducing angiogenesis, resisting cell death, and then this cancer cell is carrying metastasis at the end of this cycle. But this picture was drawn in 2000, in the earliest 2000, 2000, 2001. Then we're gonna see another article written by Douglas Hanahan again 10 years later. But first, we're gonna see what these hallmarks are in the body. Sustaining proliferative signaling. So growth promoting signals, somatic mutation activate additional pathways. Disruption of negative feedback mechanism and proliferative signaling can trigger cell senescence. What means senescence? Senescence is when, um, how can I say, when are getting old, you are getting aged, you are aging, you are getting old. So the cell is getting older, but cancer avoids these mechanisms. Choose a moment. I'm gonna change what of diets, uh, how this mechanism works. There are some um, not chromosomes. There are some genes or proteins transcripted by the genomic in the cell. So there is a conflict every time from getting a war between tumor suppression proteins and oncopromoting proteins. Oncopromoting proteins, you can see it later, RAS, KRAS, MYC, and many others, maybe 100,000 of them. But I'm just explaining you the most common ones. And then the tumor suppression genes are going to be, as an example, just the most common ones, BRCA1 and BRCA2 for breast cancer, pancreatic cancer, colonic cancer, and sometimes brain cancer. Um, PTEN and retinoblastoma associated is the protein RB protein and PT53. These both proteins uh, have Their job is to make checkpoints of this when the cell is getting transcripted. As in the cell division, you have to duplicate the genome, you have to duplicate the DNA. So in the process, in the pathway of duplication, the DNA, you can see many mistakes on the copying the DNA. So PT53 and RB protein are checking every single of those mistakes, and their function is okay. There's a mistake. Is if that's repairable, we are going to repair the mistake and we are continuing with the cell cycle. But if that's not repairable, that's so bad, that's so big, that's something, anything, they decide to start crisis in the cell. So crisis means apoptotic. Apoptotic begins in the mitochondria. We're going to see later the mitochondria. So then, um, growth promoting signals. For example, they create new receptors, new proteins that establish the cell to grow. That motivates the cell, that enhances the cell to grow bigger, 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 and bigger. In a normal cell, uh, it will not grow so fast, it will not grow so big. So, or in a cancer cell, there is not stopping. There is no protein that says, hey, stop. So it grows a lot. Somatic mutation activate additional pathways. That's what I say about the DNA. Disruptions of negative feedback mechanisms. In the topic of physiology, you can see many negative or positive feedback. For example, with endocrinology system, there is a hormone in the brain that stimulates another hormone, another organ to create new hormones. But when there is a lot of this hormone, so okay, it's good enough. Stop the cycle. That's a negative feedback. But when there is a disruption of the negative feedback, what causes? It, it causes to continue producing, 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 no matter how big quantity of that there is in the body. It's going to be producing, 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 and again, and again, and again, and there is no stopping. So that's one step of cancer to be so effective for killing people. Proliferative signaling can trigger cell senescence. Uh, this proliferative can making a cell to get exhausted, very irritated, and then get senescence. They that gets really old and die because of apoptosis, of because of any other mechanisms like I just forgot the word in English. When you stop eating, you get inanition, I guess in English, inanition. So the cell dies itself. So evading growth suppressors, that's exactly what I told you about inhibiting RB protein and PT53 functions. So the RB is checking the DNA, copying very well, very well, very well, and then he sees the mistake. He tries to repair. If that's repairable, okay, continue with the cycle. If that's not repairable, sorry, if that's not repairable, that gets that induces the mitochondria to get in apoptosis. And PT53 is just relaxing, but when there is crisis, when there is a cell, there is a very high level of stress. The cell, the PT53 takes takes the decision to die. So we are gonna die. We are going to kill the cell. We are getting into apoptosis, and everyone is dying. This cell is getting bad coping, so we have to stop it killing ourselves. Corruption of the tumor growth factor beta pathway promotes malignancy. As you can see in the topic of immunology, growing factor means of tumorogenic growing factor means uh, the a tumor to grow. That helps the cells to grow up once they are divided into two very little cells. So this interleukin helps the cells to grow, but when the pathway of TGF beta gets disrupted or corrupted, there is no a negative feedback of the TGF, so the TGF makes a malignancy. What's malignancy? When there is a specific types of cells in this cancer. Uh, don't forget, there are many types of cancer in many types of cells or and tissues from many types of organs. We're going to see that later. Evading the cell death, apoptosis, autophagy, and necrosis. That's the three ways a cell can die. Apoptosis starts on the mitochondria. The mitochondria receives a signal from PT53, from RB protein. Um, they tell mitochondria, okay, this is not good. We are going to die. So mitochondria, the mitochondria breaks the the wall of the mitochondria, the membrane of the mitochondria breaks. So the components of the mitochondria gets to the cytosol, and in the cytosol, lysosomes, ribosomes eat the mitochondria that get kind of intoxicated, and so the whole cell die. Then autophagy, the cell starts to kill not the membrane, not the mitochondria, but it starts eating itself. Lysosomes, ribosomes, peroxidase starts to kill the nucleotides of the organelles. All the organelles in the cell start to be eaten by itself. So the cell obviously die, and necrosis has pro-inflammatory and tumor-promoting potentials. We have learned through many years, even decades, that necrosis is uh, like dead tissue. So if that's black, that's dead. Okay, but that's not exactly dead. That's exactly anoxia. When you have not enough oxygen to get into the cell, there is not enough ATP, there is not enough um glucose, there is no, the, the cell enters to a cycle of starvation. So the starvation makes the cell to get into necrosis. There is no oxygen. If you despite the cell, we need just a few milligrams of oxygen, just between 1 and 3 milligrams per millimeter of oxygen. But how oxygen works? What oxygen is useful for? Oxygen helps to mitochondria to create new uh, mm, adenosine ATP. I just forgot the name of the complete molecule in English, but helps making ATP into ATP. So the ATP is the first element that gives energy to the cell. So the cell has no energy, and then the cell dies because of starvation. But necrosis has pro-inflammatory and tumor promoting potential. Why? Because when a cell starts into necrosis, all the macrophages, all the white cells have to who try to, how can I say it, to eat the cell, the the garbage of the cell. There is useless now, so the white cells have to eat the cell to destroy and eat the cell. But that immunology system has many pro-inflammatory tissue, and the pro-inflammatory interleukins pro-inflammatory cells, interleukins, kinases, all the pro-inflammatory system helps cancer to grow because we are going to see that in a more specific way. But pro-inflammatory systems help cancer to grow. That's why inflammatory diseases like obesity, kidney diseases, heart disease, obesity, they all are riskable to have cancer. So enable replicated immortality. What it means. So, for example, as you can see in molecular biology, you can see the telomeres are the further place or complement of the chromosomes, as just the type, just very far from the center of the chromosome. So, but they are each time a chromosome replicates, divides itself, the chromosome gets short, the telomeres get shorter, 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 shorter. So there is a time that the chromosome cannot duplicate anymore. So it starts to getting into senescence and it dies. It's getting really old and die. So DNA polymerase protect telomeres. How? Is you can see the examine of the nucleotides getting into the telomeres because of the DNA polymerase. DNA polymerase is an enzyme that some people use very often in the laboratory, but in the DNA, it helps adding adding nucleotides to the telomeres. So in a normal cell, 10% of the cells, normal cells, um, DNA polymerase does not exist inside of these cells. But in the 90% of the tumors, benign tumors, malignant tumors, exist DNA polymerase who is adding nucleotides to the telomeres and then make it's is making the life of the chromosome uh, bigger and bigger and bigger. So that's why it can multiplicates many times more than a normal somatic cell. That's why that's the mechanism how cancer enabling or enables replicative immortality. That's why they reproduces so big, so fast. So the life to the telomeres may both limit and foster neoplastic progression. So that don't take this last paragraph so seriously because this is some discoveries in the lab. Of course, they have seen that really low levels of DNA polymerase can help the cancer to grow, but this is just a discovery in the lab. That's not what I'm trying to explain you. So how well, I have to explain what I'm saying. DNA polymerase, when there is any really low levels, the cell has to look for another times or for another enzymes that helps telomeres to grow, and the cell starts to grow really fast because the cell know it's not gonna last too long. So that's why they have to replicate itself really fast many times. Inducing angiogenesis. What's an angiogenesis switch? There's many. Do you remember what I said about oncogenic tumor suppressing proteins? That's exactly the same. There are pro-angiogenic and anti-angiogenic genes and consequently proteins. So these both proteins activate or fight once against another to to create or to stop angiogenesis. But the cancer makes some changes in the DNA. So many of the cancer have mutations in the anti-angiogenic proteins. So pro-angiogenic proteins are getting stronger. So that's why they make angiogenesis. Otherwise, for example, in some experiments, you can see uh, don't uh, don't put too much oxygen in a newborn's baby. Why? Because the retina, the eye, is getting too much oxygen, so it's not producing angiogenesis, and then otherwise they are getting loss of angiogenesis, and they can lose the ability to see. They can get blind. I'm sorry. Uh, hypoxia. Hypoxia can generate angiogenesis because if the tissue detects that there's not enough oxygen, then they start or the tissue starts making angiogenesis to receive more blood, and the blood gets more oxygen, provides oxygen. What's the pyramid? What's the center of angiogenesis? Vascular endothelial growing factor can be upregulated by hypoxia or angiogen signaling. So both mechanisms, both pathways can regulate vascular endothelial growing factor. I think the name of this contraction explains the, the function very well for itself. Vascular endothelial growing factor. Uh, well, that's clear. And gradations of the angiogenic switch that make different steps that explain the fight against pro-angiogenic and anti-angiogenic switches. Uh, what I was about to tell you about the vascular growing factor, they can be regulated by those systems, but then we have to change to another. Activating invasion and metastasis. The epidermal mesenchymal transitions program broadly regulates invasion and metastasis. That's why, because the epithelial mesenchymal is the only way to avoid cancer to get through metastasis. Once, if you see the pathology of cancer, once the cancer gets through the basal, the membrane on the base, it gets invasor. Cancer invading new tissues. So the vessels, the capillaries of the cell are on the other side of the membrane in the base, or the basal membrane. I don't know exactly the name of it in English. But once it gets through the membrane, it get access to the sorry, to the capillaries of the tissue, and they can through uh, another further organs very far from the origin cancer, thanks to the vascular system. Distinct forms of invasion may underline different cancer types. It totally depends on which cell was stimulated more. Um, that depends on the risk factors. You can see a glandula can have many types of cells, and it depending it's or depending on which cell is getting activated the most, is stimulated the most, it depends on which cancer is going to be. There is enormous complete Bible about the types of cancer. If you remember, there are many types of cancer in a simple cell, many types of cancer in a different tissue, many types of cancer in a different organ, from many types of different systems, organic systems. So that's what it means when distinct forms of invasions may underline different cancer types. So different ways of the access to the basal membrane. This schema represents on how the um, represents on how the hallmarks of cancer make cancer. So the changes in gene expression, if you can see every time every differentiations ways of proteins are getting to changes in gene expression. If you can see, you can see here P53 is the DNA damage sensor. Then you can see in the other way in the proliferation circuits RAS, MYC, TCF4 that represent e-gathering. It represents how the proteins alters the system. But if you just try to forget just all the screen, all the lines of all the circles, uh, the motility, the cystostasins, the proliferation and viability circuits, all of them conduces to a center which is changes in gene expression. If you remember what I just told you a few minutes ago, all the cancer, all what cancer represents is changes in the DNA or damage in the DNA. So all the hallmark capabilities of cancer are here in changes in gene expression. What I told you about, there is a fight into suppressors and promoting signals, and so cancer has to kill or the sequencing of the good ones. So that's why the promoting signals wins that kind of fight. Um, I don't see RB. Maybe this is the PRB. Uh, DNA damage sensor, of course, this is the most important one. This is the PT. This is PT 53. BCL2 represents about apoptotic. Apoptotic start into a very grown family about BCL2, BAX, BAK. There are many cytokines that helps the mitochondria to start into the apoptotic pathways. So that hallmarks that I just told you before were written in the year of 2000. 21 years ago, was were written, but in the year 2010, four new hallmarks were established. If you can see here two, three, four, five, and six. The six in the origin. And then there are two emerging hallmarks. Sorry. There are two emerging hallmarks trying to avoid immune destruction and the regulating cellular energetics. And then that's how two new hallmarks helps the cancer to appear in the body. And then two new hallmarks that helps cancer to establish itself in the body and not to be killed by the body. That is genome instability and mutation. That's what I'm trying to tell all this time. And two more promoting inflammation. As I told you, inflammation promotes signaling to tumor to the growing of the tumor. So enabling characteristics, the two ones I told you, genome instability and mutations, it could be resumed in just this amount of genes or proteins. Uh, we could write a book, a complete book of any one of these with many volumes, but we are just going to mention the first or the most important ones. P10, the function of PTEN is a suppressor to a growing factor, a tumor suppressor protein. So if P10 gets um, depleted, So that's why people start to making tumors. I once saw a patient uh, 2 or 3 years ago with tumors in the canal of the hyric, tumors in the neck, tumors in the tongue, tumors in the penis, tumors next to the nails. So they are making tumors everywhere, and some other tumors. If you have tumors, you have a lot of risks to get cancer. Of course, that patient had five different types of cancer before. That's a mutation in PTIN. BRCA1 and BRCA2. The most important is BRCA2 because it helps to the in the cell cycle, in the cell division. It helps to cut the DNA. So because the DNA gets rolled itself into two cords are getting rolled. So if you are pulling so hard, it gets really tight, really tight. So that's why the BRCA2 gets cutting into pieces. So it's gonna be easier to separate the binds of the cords of the DNA, the two complements. It's gonna be easier to separate them. PT53 is a checkpoint, KRAS, NYC, and PAKA. PAK3CA that represents pro-oncogenic factor. And tumor promoting inflammations, it means like lymphocytes, especially T, CD4, T, CD8, and the lymphocytes natural killers. So that's why making inflammation makes interleukins that make more inflammation of the system, and the inflammation helps cancer to grow and to establish. So that's why tumor promoted inflammation makes an enabling of the cancer characteristics. So this is a cycle of how this pro-oncogenic factor disrupts the P53 function. So the P53 is the guard of the DNA. He's just checking everything is going okay, and then get the NYC cycle and stops the function of P53. That is a kind of cycle you can see um, about the topic of cancer metabolism. This is not the topic of this class, but this is very important, the cancer metabolism. And this here, emerging hallmarks, reprogramming energy metabolism, and the meaning of glucose metabolism, glutamine metabolism, and fatty acid metabolism. The, if you can see what I told you a few moments, um, the cell could start in senescence, the cell could get into apoptotic, into crisis, into necrosis, even necrosis. So the cell is changing. The cancer cell has to change the metabolism, mainly the energy metabolism of the cell to survive. So if the cancer is getting damaged because of the cancer, it's getting into hypoxia, um, into starving of nutrients, so the cell could start in apoptotic. So what cancer has to do is to repair glucose metabolism, especially glucose metabolism, to get enough uh, energetic systems to survive. And evading immune destruction, I'm going to show you a video after that about, and this picture where was so big, so I have to separate the picture into two pictures. And this is the glucose metabolism. If you can see here in the top the glucose, then you get glucose, and then you get glucose 6-phosphatum, uh, or phosphatate. I don't know exactly the name in English, but it's glucose 6p. Let's gonna name it 6p. And then NADPH, NADH, and then ribosomes, ribose 5P. I don't know to say pentoquin. Um, 5P. Let's gonna name it 5P, ribose 5P, and then forming a nucleotide. So if you see the red or the molecules mm, uh, signaled with red, you can see the tumor suppression factors or tumor suppression proteins, P53 mainly in this picture. And if you see the proteins marked in blue or kind of blue, you can see the oncoproteins AKT, NYC. I saw KRAS here, so they affect the cycle of the glucose metabolism, trying trying to avoid starving and trying to survive as a cancer cell. So in the other side of the picture, we could see the mitochondria. In the center of the mitochondria, inside the mitochondria, gets to the Krebs cycle, to the Krebs to acid cycle, many names of this cycle. But if you can see, glutamine metabolism is also altered by micro R23. Here is KRAS. I saw it here. KRAS, NYC, all of them AKT, PI3K, all of them uh, trying to affect or to cooperate with the metabolism. Fatty acid, glutamine metabolism, as we saw in the last scene, uh, glucose metabolism. So this is how the way the metabolism is affected by oncoproteins. Oncoproteins is the only way are cooperated with tumor suppression because they are trying to restore, trying to save the energetic mechanisms. Diagnosis and prevention. Why I quote prevention here? Because the diagnosis is the only way that you can have that you can prevent about it. Many articles write about magnetic resonance, tomography, polymerase chain reaction, DNA sequencing, and other for specific cancers. For example, for prostate cancer, you can ask for a prostatic specific antigen, prostatic specific antigen, C819, CA19,5. There's many of tumoral markers. But here in prevention, for example, I told you something about hereditary cancer. If your if your grandfather died on his 40s because of pancreatic cancer, then your mother got breast cancer at the age of 30, and then your sister got cancer at the age of 30. Uh, you this highly an enormous risk that you and your family have hereditary factors on the DNA, on somatic cells. That's why you have to make a DNA sequencing, then establish a mutation in the DNA in one of those tumor suppressions oncogenes, tumor suppressions proteins or oncogenes or oncoproteins. If you have one of those mutations, then you can establish a phenotype of polymerase chain reaction, and then you can study all of your family. And that's why, for example, there is a very extremely solution, but that's the actual solution. If you have the Angelina Jolie's effect, uh, she discovered that she had some risk factors on her DNA, so that's the surgeon perform a bilateral mastectomy to trying to prevent cancer. So that's the idea of the new treatments of cancer, of the new mechanism of prevention from cancer. That's DNA sequencing, polymerase chain reaction to establish a different mutations into DNA patients, and then try to take the organ before um, cancer takes it. Uh, not so extremely, but for example, um, if you have risks to perform pancreatic cancer or liver cancer, then you can, for example, make a magnetic resonance once a year, trying to prevent and try to discover cancer at the beginning, not at the end. That's what I'm trying to tell you about the promotion of the health system in Mexico. They tell you, if you have the skin of the breast like an orange, if your nipple gets inverted to the other side, if you can see a tumor, a visible tumor, uh, that's late. That's extremely late. Uh, the cancer is advanced, maybe with metastatic brain or something like that. So we have to change the topic we have to change in our minds the switch of prevention. The prevention should be um, DNA sequencing, for example. So these kinds, we are talking about new treatments. This new treatment is going to be the future. But prevention with genomic, it's the future of the future. If you could start this from now, we are saving maybe millions of people from cancer. But then there is a last screen about the treatment. There are three pillars in the cancer treatment. The first one is chemotherapy, the chemo, then the surgery, and then the radiations of the restant tumor from the surgery in the body. But that's really old. For now, there is a new treatment that is immunotherapy. Why immunotherapy? Immunotherapy, uh, it's the treatment is the future. Of course, it's the future. Actually, Dr. Dr. Paul Janssen Allison won the Nobel Prize in 2018 in medicine for discovering a new kind of treatment for cancer, which is TCDLA4 receptors. TCDLA4 receptors, uh, there is a mechanism on which lymphocyte touches the cell and tries to destroy the cell, but they go further. They get filtered and they die because of this receptor. So Dr. Paul Janssen Allison discovered a new supplement that avoids the receptor, so lymphocytes proliferate comfortably and they could destroy the cell. So for last, I'm gonna show you this video, and then we could start with the questions, with the question of the oncogenesis. Um, Sorry for the resolution of the video.
It doesn't make any difference what the cancer is. You're treating the immune system, not the tumor cell. This could serve as a therapy for treating all cancers.
James Allison is an immunologist, a seasoned harmonica player, and the scientist behind one of the greatest advances in cancer therapy in the past 15 years. He is the winner of the 2018 Dr. Paul Janssen Award for Biomedical Research and a champion of science. It was immunology, not cancer, that drew Allison into the lab. In the 1970s, scientists did not know much about immune cells. He spent 15 years teasing out the basic biology of killer or cytotoxic T cells. These cells identify and destroy threats like viruses, bacteria, and tumors.
I said, I'm gonna start working on these, someday and figure this stuff out.
Allison found how a cytotoxic T cell identifies, binds to, and destroys a tumor cell. At the molecular level, a T cell receptor binds to a highly specific protein complex on the tumor cell, activating the killer response. At the same time, a different receptor on the T cell binds to a molecule on the tumor cell. When both receptors are bound, the T cell starts to proliferate madly. The goal is to make enough T cells to attack and kill all the available threats.
It's kind of like flipping the ignition switch in your car. You're not going to go anywhere until you press on the gas pedal.
But that gas pedal can only be pressed for so long. Eventually, T cells slow down and
Anyway, they just...
work by other scientists caused Allison to look at another receptor called CTLA4, which slows down T cell division. It's kind of like brakes against the gas pedal.
We call it a checkpoint, because it's something that stops division.
The checkpoint prevents T cells from proliferating indefinitely. But if it occurs before T cells have eliminated all of the tumor cells, the tumor might win.
If we could just disable the brakes and let the T cells keep going, then we can make the immune system always win.
The idea of checkpoint blockade was born. In the 1990s, Allison developed an antibody that would bind CTLA4 and disable the T cells' brakes. In the early 2000s, the anti-CTLA4 antibody was developed into the drug ipilimumab, IPI for short. Clinicians first tested it on 14 metastatic melanoma patients with nothing to lose.
The median survival after diagnosis was 11 months, and no drug had ever improved on that.
In that first trial, meant only to test for safety, 3 out of 14 patients showed tumor shrinkage.
And so that caught everybody's attention, because that just didn't happen.
He still remains in touch with early patients.
One of them actually, she had metastatic melanoma. She failed everything. That was 18 years ago. She's got a vibrant family now.
IPI turned a previously untreatable cancer into one that could be cured in 1 out of 5 patients. Now, many checkpoint inhibitors and modulators are on the market.
Seems that there's a new one coming every 3 weeks or so.
Combining IPI with another checkpoint drug ratchets survival rates up to 60%. That's good news for patients. But what about the other 40%? Allison thinks the answer can be found by going back to basics.
When you're a basic scientist looking into fundamental mechanisms of biology, you're doing it because you want to understand how things work. I really didn't get into any of that work to do anything about cancer. To do that and have it turn out that it's something that actually helps people is something that's very rare. And I'm really lucky to have had the chance to do that.
Okay, this was the last screen of this class. Just like an extra commentary, I would like to say that this is a phrase that really motivated me many times, that we have to go by basics to understand and try to beat the future. Now, if you have any questions, you can do it whenever you want.
Thank you so much, doctor. And you can listen to me because okay,
yes.
thank you. So thank you so much, doctor. Very interesting webinar. So I don't know if anyone has one question, you can write it down on the chat, or remember, you can open your microphone and do the question. While we are waiting, if someone wants to ask for something, I would like to ask you some questions, if you don't mind. One of the questions that I have for you, doctor, is I don't know if the consumption of vitamins, of some nutrients, can affect the pathway, the metabolic pathway. They can increase the cancer, because there are some doctors that when some patients have cancer, they don't allow them to eat some kind of vitamins or minerals or something because they say there might be a risk of increasing the cancer or the metastasis and all stuff. So I don't know if you know something about it.
Uh, maybe in some kind of tumors, for example, in tumors in endocrinological system, they produce many. What's the name of that? Uh, Tumoral factors
Mhm.
through the nutrients. There's used a hallmark, but if you try to make the tumor into starvation, you are gonna kill the whole system. So uh, there is a clearly reduction of some specific vitamins for trying to stop the upregulating of the growing cell, but there is a mechanism when the cell starts to get into apoptotic mechanisms. So that's why the cancer system stop apoptotic and modifies the um, the glucose metabolism, for example.
Okay, thank you so much. Another question, if you don't mind.
Okay.
Here in Mexico, what about the prevention? What are the politics in cancer? I mean, the public politics about cancer. What are the prevention factors or the prevention mechanisms that we have here for a patient with cancer?
Um, the public prevention, actually, there is nothing. There's absolutely nothing. Just the prevention the government gives to the population is just try to eat healthy, do exercise, get into a walk for 30 minutes a day, and eat healthy. Don't smoke. Don't drink too much alcohol, and don't be don't be fat. That's the only prevention for everything.
Okay, and what about the detection of cancer right now in the public hospitals? Well, which one would be the PCR of, I don't know, clinic labs, or I don't know what are the normal pathway or the normal path to do to diagnostic the cancer.
Okay. The, the patients could go to the first level of health attention. Then, for example, in breast cancer, they practice some mastography. There are some kind of questionnaires that's that's pretty disappointing. That's disappointing because the doctor realized 5 questions, and with that 5 questions, for example, do you pee more than 3 times a day? If the patient answers no, okay, you don't have prostate cancer. That's that's something for 40 or 50 years ago. That's not very um, clear.
Okay. The question is because I know prevention is one of the biggest step to diagnostic the cancer, they prevent the cancer, no? And I have another question. What is your suggestion about the measure of about a gene and the gene expression? I don't know, a protein, or which one would be the one that you can suggest us to do before to prevent the cancer, the metastasis on any stage of the patient?
Yeah, okay. My what I would recommend is I don't know the exact name in English, but for example, when the baby is born, they apply in Spanish el
Tamizaje.
tamiz, el
Mm.
tamizaje. So that tamizaje will be, for example, a DNA sequencing. That would be extremely expensive, but that could be the only way that people know for, some years before they could have cancer and they could prevent. But the prevention it's extremely. For example, uh, if you have mutations in BRCA2 genes, the recommendation is okay, go to the doctor and apply bilateral mastectomy today. So that's extremely, I think.
Okay, thank you so much. I don't know if any guys have a question for the doctor because it's really interesting to know his opinion about this topic. Or you can open your microphone, you can write it down. I don't know. So, oh, everything it was well understood because it really was. It was a really great chat. So you don't have any questions? Okay, guys, if you don't have any question, I would like to thank to Dr. Machain. It was a really interesting webinar. I learned a lot, and I hope the student has learned a lot too. So thank you so much for being here, and I hope the next semester you can join us with another webinar. And if you
Hopefully.
don't go, of course, but if you stay here, I would like to invite you again so you can join
Okay,
us
of
and give us
course,
another
of course.
topic. And and you can have this topic. The next topic can do it in English or in Spanish. We have the two version now, so
Okay.
it's gonna be great if you can join us. So thank you so much. I would like to recall you that tomorrow our webinar is in Spanish. And that webinar, the topic is going to be manejo farmacológico y nutricio del paciente con diabetes mellitus with the Dr. Ariel Roldan Mercado Sesma. So I hope you can join us. Tomorrow is going to be at 7 o'clock. You know, the same channel, the same everything. So thank you so much for being here, guys. Thank you so much, doctor, for joining us and give us all your knowledge about this topic. And I hope you can see you really soon.
Hopefully, and thank you for the invitation. I would really love to make another webinar in the next chance.
Okay, thank you so much. So good night, guys. Have a great
Thank
dinner
you.
and have a great rest. See you. Bye.
See you. Thank you.